Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Fitria binti Ahmad Munir and my matrix number is 23DBT 20F1036. Before I start the simulation, I want to explain about the theory or background about SDS page. SDS page is stand for sodium dodecyl sulfate polyacrylamide gel electrophoresis. What is SDS page? SDS page is a method of separating proteins based on their molecular mass. Polyacrylamide gel is used as the gel matrix for separation. SDS is important role in protein gel electrophoresis. SDS also strong detergent, present in high concentration in the buffer that prepares samples for electrophoresis. Before samples such as cell can be run on a protein gel, SDS need to lyse cell membrane and solubilize all proteins. Purpose of SDS is: first, breaks the non-covalent bonds; second, binds to the amino acid side chains, giving the protein a net negative charge. So that is the page gel diagram. Page gel is divided by 2, which is taking gel and separating gel. Okay, I can start the simulation experiment. Okay, this is the protocol of this experiment. So this experiment have 6 protocol and start the simulation. Okay, first collect cells, increase the heat block temperature to 95 degrees Celsius. Okay, set for 95 degrees Celsius. Okay, after that set a micropipette to 30 microliters and attach a tip. Okay, set for 30 microliters and save the volume, then attach a tip. Okay, then draw up the wild type cell culture. Close the wild type cell culture and dispense the wild type cell culture into the wild type cells tube. Right after that, eject the tip into the biohazardous waste container to prevent contamination. Okay, next, repeat the procedures to transfer 30 microliters of the pancreas cell culture. First, attach a new tip. Then, draw up the pancreas cell culture. Then, dispense into the pancreas tube. Right, eject the tip into the biohazardous waste container to attach a new tip. Alright, then draw up the edit for cell culture. And dispense into the edit for tube. Right, done. Eject the tip into the biohazardous waste container and Then attach a new tip to repeat the procedures. Now draw up the edit 7 cell culture. Then dispense into the edit 7 tube. Right, done for protocol number 1. Okay, don't forget to eject the tip. Alright, next. Second protocol is add a sample buffer. Set a micropipette to 10 microliters and attach a tip. Okay, set for 10 microliters, then attach a tip. Okay, attach a tip and draw up 10 microliters of 4 times SDS sample buffer. Right, draw up. Don't forget to close the 4 times SDS sample buffer. Then dispense the 4 times SDS sample buffer into the wild type cells tube. Before repeat the procedures, eject the tip into the biohazardous waste container first and attach a new tip. Okay, done. Okay, draw up the 4 times SDS sample buffer. Then dispense into the pancreas cells tube. Okay, as usual, eject the tip to prevent contamination. Then attach a new tip. Next, draw up 4 times of SDS sample buffer. Then dispense into the edit 4 tube. Okay, next repeat the procedures. Attach new tip. Right, draw up 4 times of SDS sample buffer. Then dispense into the edit 7 tube. Right, eject the tip into the biohazardous waste container. Okay, done for protocol number 2. Next, denature the proteins. First, move all the 4 sample tubes to the heat block. Right, pancreas tube, edit 4 tube, and lastly edit 7 tube. Right, set the heat block timer to 10 minutes. And press start. Right, after that, remove all the samples tube from the heat block. Okay, remove. Centrifuge the samples. First, move the samples tube to the microcentrifuge. Alright. Okay. Next, check the balancings. Make sure balance. Right? Okay. Then close the lid. Then press pulse to spin the tubes. Then after that, remove the tubes from centrifuge. Drop the tubes into the rack. Alright, done for protocol number 4. Okay, close the lid and next, load the gel. First, place the polyacrylamide gel in the electrophoresis box. Then fill the gel cassette with one-time running buffer. Make sure in the gel cassette and remove the comb. Okay, typically precast gels come with a comb that has to be removed before electrophoresis. Okay, remove the comb. And fill the outer gel electrophoresis tank with 1 times running buffer. Okay, done. Set a micropipette to 20 microliters and attach a tip. Okay, set for 20 microliters. Save the volume and attach a tip. Right, draw up the protein ladder. Then move the micropipette to the first lane of the protein gel. Okay, pick number 1 and next dispense that. Next, dispense 20 microliters of the positive control into the second well of the gel. Okay, first draw up the positive control. Then dispense into the second well of the gel. Right, eject the tip. Attach a new tip. Okay, next, dispense 20 microliters of the negative control into the third well of the gel. Okay, draw up the negative control. And dispense into the third well of the gel. Alright, dispense. Done. Okay, as usual, eject the tip into the biohazardous waste container to attach a new tip. Now draw up the edit for cells and dispense into the fourth well of the gel. Okay, pick number 4 and dispense. Alright. Okay, eject the tip. Attach a new tip. And draw up the edit 7 cells. Then dispense into the 5th well of the gel. Alright, okay, done. Okay, don't forget to eject the tip into the biohazardous waste container to prevent contamination. Okay, next, cover the electrophoresis box. Then connect the red and black cables to the power supply and the lid of the gel electrophoresis box. Black with black, red with red. Okay, next turn on the power supply. Then set the voltage to 150 volt. Then set a timer to 60 minutes. Okay, then start the power supply. Wait until the time has elapsed. Okay, turn off the power switch. Unplug the electrodes. And lastly, inspect the gel. Eject the gel cassette. Alright, that's all. Thank you.